So I mentioned just now, okay, it is more on the experiments. Yeah, so what are the terms that you need to know? What are the terms you need to understand? How to conduct a fair test and how to, to identify the aim of the experiment. Yep. So to recap just now, okay, I asked about the condition of the germination, right? Then what is the purpose of seed dispersal? What is the purpose of seed dispersal? Is seed dispersal for ensuring the continuity of its own kind? Is that the answer? Is reproduction, okay, is seed dispersal ensuring the continuity of its own kind? So what do you think? No, okay, seed dispersal is not to ensure the continuity of its own kind. Remember that seed dispersal is to prevent what? Prevent overcrowding. So why? Okay, why do the plants need to prevent overcrowding? So why? Okay, why do they need to prevent this? So that the plants would have less competition. Yeah, so of course, okay, the further they are from one another, the less the competition would be, right? So then my question is, what are they competing for? So there are 4 things which they compete for. Will we get the free bubbles at the end? If you answer my questions, okay, if I see your effort, then okay, I will reward you with this free bubble, okay, at the end of the lesson. So what are the 4 things that they compete for? Sunlight, water, then space, and mineral salts. Okay, how about air? Do they compete for air? No, they are not competing for air. Why? Because air is everywhere. Air is readily available. So they are not competing for air. So they just compete for space, water, sunlight, and mineral salts. What is the difference between ovule and ovary? So ovary is the outer part. Okay, so the part where it contains ovules inside. So ovules are always found inside the ovary. Now, my question is related to this. After fertilization, which one turns into fruit? Okay, so after fertilization, so there are 2 things that happen. So ovary becomes fruit and ovule becomes seed. So like watermelon, okay, so are there many seeds inside a watermelon? So can you find a lot of seeds inside a watermelon? Yes. Now my question is, inside the ovary, is there only one ovule or are there many ovules in the flower of watermelon? Yes, okay, so if you see that the fruit has a lot of seeds, it means that the flower, okay, the ovary, okay, to be more precise, so in one ovary, you would find a lot of ovules. Why? Because remember, ovary turns into fruit while ovules turn into seeds. Yeah, so good job. Yeah, so you guys can recall most of the things that we have learned so far about the reproduction in plants. Now, then let us move on to our main topic today, which is the experimental based questions. Now, to understand and to know how to answer these questions, the questions which are related to the experiments, you need to understand the terms which are always involved and used in the experiments. like the aim of the experiment, the hypothesis, variables, fair test, control setup. So you need to understand these terms. So beginning with the aim of experiment. Yeah, so the aim of the experiment, like the name states, it is to state the goal, the purpose of the experiment. Usually, right? Okay, so for the aim of the experiment, there are certain keywords that you can look out for. So let's say, okay, you see a question related to the experiment. So how to identify the aim of the experiment? What is the keyword that you need to look out for? What is the keyword that helps us identify that it is the aim of the experiment? So the keyword to identify, it is to find out. Yeah, so this is the keyword for aim of the experiment. Yeah, some of the aims of the experiments begin with to investigate. Yeah, to investigate if blah blah blah. But I think for most of the questions I came across, they usually put to find out if. Yeah, so to find out if. So that's why if you are to understand and look out for the aim of the experiment, please look out for this keyword, to find out. So then the second one is the hypothesis. Joe, what is hypothesis? So you can see okay, the sentence explaining what hypothesis is. But to summarize what hypothesis is, I can put it in this way. Hypothesis is like prediction. You observe something and then you predict. That is a simple term of hypothesis. But of course, in science, right, you cannot make wild guess. Can you? Can you make wild guess? Like you guess anyhow? No, right? Like you observe something. Okay, so okay, you just do a wild guess. I mean, you can't. Of course, okay, when you observe something, then based on your prior knowledge, based on what you have seen so far, based on what you have learned so far, then you could make the scientific prediction. So that is what hypothesis is. For example, so I see that the plants, right? Let's say the water plant. Okay, so I have water plant and I have lamps with different color. Then I observe that the plants, right, release different amount of air bubbles under different light. So based on this observation, then I can make a prediction. I can make a prediction. Oh, based on what I know, I think that blue light will allow the plant to produce more air bubbles. Okay, because based on what I know, chlorophyll. Okay, so the green pigment in the plant, capture blue light more than other light. So that's what hypothesis is. It is a prediction, but it is not just a wild guess. It is a scientific prediction based on what you know, based on the prior knowledge, based on the established knowledge as well. Yeah, so that is what hypothesis is. So another example, so you can see in the slide, Alan thinks that natural fertilizer works better for his plant. So he planned an experiment to confirm this. So he wants to compare which one is better, natural fertilizer or synthetic fertilizer. So then after that, then he creates the aim of the experiment. Yeah, so the aim of the experiment is to find out or to determine whether the plant grows taller or looks healthier, yeah, it depends on what he is trying to see. Okay, with fertilizer A, which is the natural one, or fertilizer B, the synthetic one. Yeah. So then, so the hypothesis. So based on the prediction, okay, so based on what he know, most, okay, so like usually, right, okay, natural things is much better than the synthetic ones, right? Yeah, so based on this prior knowledge, then he makes a prediction, which is the hypothesis. So the plant grows taller in the presence of fertilizer A, which is the natural fertilizers. What is synthetic? Synthetic means that it is made in the factory. It is made by humans. Natural fertilizer usually is like from the animals dropping. So that is natural fertilizer, like the cows, okay, the cow's dung, okay, the buffalo's dung. Yeah. So then, yeah, okay, so that's how to differentiate or understand the aim of the experiment and the hypothesis. So any question regarding this? Okay, so and no questions? Okay, then let us then try this together. So I want you to understand first before you try answering this. Okay, so the next variable, okay, the next term that I want you to understand is the variable. Like, have you heard change variable, measured variable, the variables that need to be kept, the variable that is always measured? Have you heard this term variable? I think, okay, most of you have heard this term before, right? Yeah. So then, what is variable? What is the definition of variable? So to put it simply, variable is the factor that can affect the experiment. Any kinds of factor that can affect the experiments is called variable. Yeah. So variable are the factors that can affect or that are involved in the experiment. Yeah, so there are three different kinds of variables. The first one is the variables that needs to be kept the same throughout the experiment. Yeah, so these variables have to be kept throughout the experiment, kept the same throughout the experiment. So we call this as the constant variable. So why? Why do we need to keep some of the variables? Or why do we need to keep the constant variable the same throughout the experiment? Anyone can give me the answer? So that our experiment is a fair test. Yes. So in order for you to have a fair test, in order for your experiment to be a fair test, so you need to know which variables need to be changed, which variable needs to be measured, and which variables need to be kept the same throughout the experiment. Yeah, so that is what constant variable is. Then the next type of variable is the change variable. Yeah, so change variable is the variable that is being changed, the value, the type, the, okay, so it is always being changed throughout the experiment. Like for example, just now, okay, the experiment that is involving the fertilizer. Okay, so which one is the change variable? Can you predict what is the change variable of Alan's experiments? Oh, very good. Yeah, so the change variable is the type of fertilizer. Why? Because Alan changed the type, right? Fertilizer A and fertilizer B. Correct. And then, what is the measure variable, which is the third type of variable? It is the variable that is being measured throughout the experiment. So what is the measure variable? So what does he measure? The height. Okay, so the height of plants. The height of plants. So go back to the fact experiment, okay, which later, okay, will be okay, in the next slide. Okay, we'll be we'll be learning this, okay, in the next slide. But to put it simply, okay, to have a fair test, you need to have one change variable throughout the experiment, while the other variables are to be kept the same. Yeah, so that is the key to have a fair experiment. Okay, so Clara asks, sure, why the height of the plants, the measure variable? So you see that the measure variable is the variable that is always being measured throughout the experiment. And we can see here that in order for us to know the effect, or in order for Alan to know the effect of the fertilizer, he needs to see the height, right? How the height changes throughout. So he needs to measure that every single day, correct? Okay, to see how the fertilizer affect the changes in the height of plant. So that's why the height of the plant is the measured variable. It is being measured. I thought it is weeks. No, weeks is the period. Okay, it is period. Oh, you mean measured in weeks? Yeah, okay, it can be daily or can be weeks. In this case, okay, it's weeks. Yeah, but okay, it has to be consistent, whether it is daily or weekly or monthly. But going back again to Clara's question, why is the height of the plant the measure variable? Because it is being measured, okay, continuously throughout the experiment. Then I want you to think the constant variable or variables that needs to be kept the same. Can you give one example of variable that needs to be kept the same throughout? Constant variable. Type of plant. Why? Why do you need to keep the type of plant the same? Because different types of plants have different growth rate, right? If you use different plants, then your result will not be reliable. You cannot trust that result. Other than that is the amount of water given. Yeah, so that one also needs to be kept the same. Why? Because if you give different amount of water to different plants or different setup, that might affect the result. Your result might not be reliable. Your result cannot be trusted. Yeah. So the amount of fertilizer, so Melody again mentioned that as well. Okay, the amount of fertilizer. Yeah, so you can think of any other variables that needs to be kept the same. Yeah, so the variables that need to be kept the same are the variables that are not change variable or measured variable. Yeah, so any questions on the variables? Oh, this one is the same thing that I asked just now. Yeah, so it is the type of plant, the amount of water given, then okay, the height, okay, in the beginning, So of course, okay, you should not have different height of the plants with different height at the start of the experiment. They have to have the same height. Okay. Then the next one is the amount of the type of soil. If you use like one setup, garden soil, and the other setup, muddy soil, I mean, the result would be different, right? So you do not know whether the result that you measured is due to the type of fertilizer or the type of soil. Yeah, so that's why you need to keep the type of soil the same as well. And can you guess what is the last one? Okay, I think some of the students already answered correctly. It's not the number of weeks, okay? It's not the period. Yeah, the period, yeah, okay, so you have to have the same period for all the setups. But then, what is more important than that? You see that the plants need sunlight, right? The amount of sunlight. So, what affects the amount of sunlight received by the plant? Or the temperature of the surrounding? What affects that? The location. Different location, different amount of light, different temperature as well. So that's why you need to keep the location the same. Yeah, so these are the examples of variables that need to be kept the same throughout. Yeah, so it is to ensure that the experiment we conducted is a fair test. Easy peasy. Okay, so can you repeat again how to conduct a fair experiment? What do you need to have in order for you to conduct a fair experiment? You must only have what? One what variable? Okay, so a fair test. Okay, only one change variable, while the rest of the variables are to be kept the same. So like I mentioned before, and I asked before, in order for you to conduct a fair experiment, you need to ensure that your experiment only has one change variable, while the other variables are to be kept the same. Yeah. So this allows that the person to draw a correct conclusion based on the result. Why? Because the changes that are being seen so far throughout the experiment is mostly due to the change variable. So we can say that change variable somehow affect measured variable. So the changes that you saw in the measure variable is mostly due to change variable. But if I don't see any changes in the measure variable, how to draw a conclusion from that? So we can just say that the change variable has no effect to the measure variable. Like for example, the presence of light in germination. Like whether there is light or whether there is no light, the number of seeds germinating would roughly be the same, right? Because light is not affecting germination at all. Yeah, so that's why, the conclusion is usually this one. Change variable affects the measure variable. Yeah, so usually the conclusion of the result is to explain this. Okay, Mr. Diaz, when is the bubble question? Okay, wait. Okay, so we need to understand first the variables and all the terms related to the experiments. Then you can embark on answering the bubble question. So would you want to answer the bubble question where you do not have any knowledge previously? No, right? Okay, so you want to get the bubbles, right? So that's why I want you to understand first so that you all can get the bubble from the bubble questions. Okay, so other than having a fair test, what do you need to have in an experiment? So this one is not really a must, but it is good for you to have one. Yeah, so what is this one? What is this you're talking about? It is the control setup. Yeah, so a lot of experiments, okay, will have, okay, a lot of experiments actually have the control setup. Yeah, so what is the purpose of control setup? Now, I want you to remember this. Because there are a lot of questions where you need to answer or where you need to, present or to explain, describe the purpose of control setup. Have you seen one question or some of the questions where you need to write down the purpose of control setup? Have you seen that questions before? Yeah, you will see a lot, okay, quite a number of questions related to the control setup. So basically, control setup is for comparison. You need to use control setup so that you can compare, okay, the result of the control setup with the other setups. So by comparing the result from these setups, right, then you can draw a proper conclusion. Then you can answer your question. Okay, so whether your hypothesis is true or false. Yep. So then let us see the example. So the aim is to determine whether the plant grows taller with fertilizer A or fertilizer B. So it is the same experiment that Alan conducted. So how would you design control setup? So let's say, okay, this one, the first setup. What would you put in the first setup? Okay, so how about the first setup? Oh, I think first setup, I think we can just add fertilizer A. Then the second setup is fertilizer B. Then the last one, which is the control setup, then we don't add any fertilizer. Yeah, so no fertilizer. Then how to create, how to set up a control setup. So basically for control setup, right, a control setup usually has no changes at all. So in this case, it's the fertilizer, correct? So control setup is to have no fertilizer at all. Yeah, so that is what control setup is. So usually control setup has no variables, has no change variable, usually. Yeah, so you need to adjust, okay, the control setup. So control setup usually has no change variable. Yeah, it depends though. It depends. It depends on the experiment. Yeah, so you can roughly guess, okay, what the control setup would be. Then, okay, let's say I have an experiment. I have an experiment. So I have two setups. Okay, one setup is pond water with fish. Then the second setup is river water with same number of fish. Then can you guess what will be the control setup of this experiment? Okay, I repeat again. I have two setups. One setup is just now what is a lake water, with certain amount of fish. Then the second setup is river water with the same number of fish. Then can you determine what would the control setup be? No water? No, the fish will die if there is no water in the control setup. No fish? Nah. I want to see how the type of water affects the fish. So then how would the control setup be? Seawater? No. Tap water? No. Fresh water? Not really fresh water, but pure water. It's not no water, it's pure water. Pure water means that there is only water inside. Yeah, so pure water would be the control setup. Okay, so another question. Okay, another question. But I think, okay, I'll save this question for next week. Okay, let us just proceed to our question. Okay, so this is the work example 1. So what is the claim? What is the claim for this? So which one would have seeds germinating? Setup A, easy peasy. Why? Why would you choose setup A? What is the explanation? So why would you choose setup A over setup B? Okay, setup A has moist cotton wool, while setup B has only dry cotton wool. Okay, so yeah, so that is the evidence. Okay, it's not explanation, okay, it's the evidence. Then how? Okay, how would you explain this? Okay, how would the evidence you provide support your claim? So the seeds would germinate in setup A. Why? Because the seeds have all necessary conditions. So you have to mention all the conditions needed for germination. So water, warmth, and air. So all these three are present in setup A, thus the seeds in setup A would germinate. Yeah, easy peasy. So in order for the seeds to germinate, and one variable is to be removed. So which variable needs to be removed while having the seeds to germinate? So which variable that is to be removed while keeping the seeds germinating? David can just remove the lamp. I mean, so because light is not needed for germination. Easy peasy, right? Okay, so any questions on experiments, germination, before we go into the bubble questions? No? So this is now the time for you to answer the bubble questions. Yeah, so I think a lot of you are waiting for this. So this is your time to shine right now. Okay, so the first bubble question. Oh, sure. Okay, so a long description with tables, with everything. So what is the first thing that you need to do? So what do you need to see first if you see a long description and a lot of information? So what do you need to see first? Okay, so you see the question, state the change and the measure variable in this experiment. That one is very easy. You can just look at this. Look at the table. Look at the table. Well, I give you a lot of clue to answer this. Yes, I want you to do your best. Okay, so what is the answer? But sure, there is number 2 there. That one is not, that one is not the answer. Lucky, the answer is 2. Yes, the answer is 2, but the 2 here is the marks. It's not the answer. So we can see that the change variable is the one that is being changed, right? What is being changed throughout? The one that is being changed is the amount of oxygen. So it means that the amount of oxygen is being changed. So why is not the amount of air? Because based on the table, it is the oxygen, not the air. Yeah, so you need to be very careful with that. Okay, so then what is being measured? So what did Jenny measure throughout the experiment? The number of seeds germinated, right? So this one is change variable. This one measured variable. So that's why the answer is number 2. Okay. Any questions for this? Which is independent? Independent is the change variable, right? So change variable is the amount of oxygen. Then number of seeds germinated is measured variable. How many people chose 3 or 4? Some people did choose 3 and 4. Okay, so what is the answer? Now, in order for you to present the relationship between 2 variables, right? So you need to identify which one is the cause and which one is the effect. So change variable is actually the cause. The cause of changes in the measured variable. So the effect is the measured variable. Yeah, so this one is the cause. This one is the effect. Yeah, so this one cause and effect. Yeah, so relationship has to be presented in that way. Cause followed by effect, not the other way around. Okay, so long description with a huge table. The first thing that you need to do is to look at the question, how to conduct a fair test. Remember, the one here is the mark. That, okay, might not be the answer. So please be careful. Okay, so okay, are you ready to answer this? You have 1 minute and 30 seconds to answer. And then after you understand the aim, what is the change variable? What is the measure variable? Then you can decide what variables need to be kept the same. Look at the aim, the condition. Yeah, so this one is the change variable. Then what is the measure variable? The number of seeds germinated, right? Yeah, so the germination of seeds. That will be the measure variable. Then everything else must be kept the same. So here Aisha used 4 different seeds, which is incorrect, right? Why? Because we need to keep the number of seeds, the type of seeds the same. The only change variable is the condition. So that's why the answer is number 1. Okay, you have 1 minute and 30 seconds to answer. What are the conditions needed for germination? Water, air, and warmth. Remember WAW. Wow. Okay, so which one is the answer? The answer is number 2. Why? Only seed C would germinate. Present, present, present. Why would seed B not germinate? It's too hot. The seed will die. And this one, seed A, cannot germinate. Why? No air. So that's why only seed C. Okay, this one no water, so cannot. Okay, so last question. Last question. Let's go to the bubble question first. So you have 1 minute and 30 seconds for this. So the answer for this is number 2. There are 2 change variables. First is the presence of light, and second one is the presence of water, right? So you can't have 2 change variables in an experiment. So like I mentioned, there are 2 change variables, presence of light. So one is without the black cover, and the other one with black cover. So that is the presence of light. And the second one is the presence of water. One is without water, and one is with water. No, remember that experiment is unfair because it has more than one change variable. But for option 4, does that tell you that there are 2 change variable or more? Yeah, because for option 4, it doesn't tell you whether there are 2 change variable or not. And having unfair experiment is by having more than one change variable. So that's why the option is 2. Why? Because we can see that there are 2 change variable. Okay.